Ethanol fermentation is a very important topic in biochemistry and in human history. We humans have been making beer and wine for millennia, and it's a really cool topic to understand, especially if you're interested in understanding how fine wines and beers are made in practice. And so, as we'll recall from our discussions on glycolysis, we ended up with pyruvate and another molecule, NADH. And what happens when we are operating in an anaerobic environment, in other words, no O2 is present, no oxygen is present, our cells and the cells of things such as yeast will have to derive ATP via glycolysis only, and the way they do that is via ethanol fermentation. If there was oxygen present, there would be the oxygen molecules will be able to act as electron acceptors in the electron transport chain and allow the mitochondria to generate huge quantities of ATP. But when that's not possible, the cells still need energy, and so they turn to ethanol fermentation. And so these two molecules that we have present when operating under aerobic conditions, pyruvate and NADH, need to be dealt with in order to, or need to be metabolized in order for your cells to continue living. And so if we recall the structure of pyruvate, we have a molecule that looks like this. It is an alpha carboxylate, which, as we'll recall from our TCA cycle, whenever we see these alpha carboxylates, we can form a CO2 leaving group, and carbon dioxide gas will leave our molecule if we have the TPP enzyme present. And so we're going to lose a CO2 molecule, and this will be a slightly reversible reaction. And this reaction is catalyzed by pyruvate dehydrogenase, or decarboxylase, sorry. And this converts the pyruvate into something referred to as acetaldehyde. And so the molecule is still pretty much the same, except now we have an aldehyde. We have this aldehyde functional group on our molecule called acetaldehyde. And acetaldehyde is further metabolized into ethanol, and in the process of doing that, we convert this NADH molecule into NAD+. And this is very important. I should probably draw these arrows more appropriately. So the NADH molecules will come in and catalyze the forward reaction of acetaldehyde into ethanol. So this aldehyde now becomes a primary alcohol, like so. And in the process of doing that, the NADH becomes NAD+. And this is very important because NAD+ is a required reactant in glycolysis. And as we'll recall also, glycolysis allowed us to generate a couple of molecules of ATP. And so if we run out of NAD+ in our cells, glycolysis stops, we lose our ability to generate ATP under anaerobic conditions, and the cell will die. So being able to generate NAD+ is very important, as well as get rid of the pyruvate molecules that are no longer needed. Now, the consequence of this is the creation of ethanol. And ethanol is commonly abbreviated ETOH, particularly by organic and biochemists who have to write these names very often. And it's a double-edged sword because these yeast cells, for example, will be able to survive, but the byproduct of their survival in the anaerobic environment is ethanol. And ethanol in high enough doses is toxic to yeast cells, and it will actually cause them to die. And so ethanol acts as a preservative in beer, for instance, and it's the reason why in a lot of in the old times, such as in Paris when the water quality was poor, people would drink wine instead of water because the high concentrations of ethanol would prevent harmful bacteria from being able to survive in the wine, and people could actually live off that. And so the main takeaway from ethanol fermentation is it allows for the recovery of NAD+ to keep glycolysis going, to generating ATP. And so this summarizes the steps involved in ethanol fermentation, and I hope you guys find this useful. Let me know if you have any questions, and thanks for watching.